From Microbe TV, this is Immune, episode number 15, recorded on December 20th, 2018. I'm Vincent Racaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hey, how are you doing?
Okay, it's the last episode of the year.
It is.
And joining us from Worcester, Ohio, Steph Langel.
Hi there. Yes, it's good to be here. Last one of the year. I think it'll be a good one. And gosh, I'll be in a new place next time.
That's right.
Exciting.
It's your last podcast from Ohio, right?
Yes. I wonder if you'll sense it when I move down there.
Hard to know, right?
Hard to know, right?
So it's been a month since our last episode. Someone on Twitter said today, when's the next Immune?
Oh, here it is.
And I said, today. Well, we're recording today. And it is Steph's turn. What are you going to talk about today, Steph?
Yeah, this was inspired by, we had somebody who wrote in an email and wanted to talk about neutrophils and neutrophils and their role in, particularly she had mentioned Staphylococcus aureus, but it's involved in a variety of different diseases, bacterial, viral, fungal, and autoimmune, and cancer, of course. And so that's the cell type I'll talk about today, are neutrophils.
Neutrophils.
Yes. Yep. Neutrophils. And, it's interesting because they are the most abundant leukocyte in mammals. I think they often get kind of cast aside as, there's so many of them there. They're not that diverse. They're not adaptive. So who cares?
So what's this name here? We got neutrophils, basophils. What other phils do we have?
Well,
eosinophils.
And can you tell us what the neut part means and the phil part?
Well, yeah, the neut part, I'm not sure. Cindy, are you familiar with the neut? I'm sure it's Latin.
Eosinophil, basophil.
Must have to do with staining, right?
I'm sorry. Yes. Okay, the staining. Thank you. Yeah. So basically, the granules inside neutrophils, they have this like trilobular nucleus. There's like 3 lobes, and the way that they stain I think it's a azure blue stain, and that's what makes this very bright contrast of neutrophils in pathology. And so, okay, yes, it would be the stain. Thank you. And so these cells, they come from our bone marrow, of course, like most, from hematopoietic stem cells. And you can make up to 1 billion of these cells per kilogram per day. So
Whoa.
It's a huge amount of cells. And
cool.
Pretty cool. And they increase exponentially, of course, during infection, or other cases would be clinically, if you have neutrophilia, it could be a sign of a potential problem. And so clinically, they're very important. Neutrophils, I remember I first became interested. So when I was a master's student, I was in a bovine immunology lab, and we were studying mastitis, which is the infection, of the udder. And of course, in humans can get mastitis. It's infection of the breast tissue, particularly if the woman is lactating. And neutrophils, when there's an infection, it's actually a test that can be used on farm to determine if your cow is clinically ill or not. And so we studied Staph aureus, and because neutrophils do some pretty neat things in the context of those infections, is when I really started to develop an interest. But I don't know, it's kind of with time, you're looking at other cell types, your focus goes away for a little bit. So it was fun to pop back into neutrophil biology, and they just do so many different things. And it was neat to, so I'm looking forward to share that with you guys.
So neutrophilia is a lot, and then neutropenia would be fewer, right?
Right. Which also clinically can be relevant if you are missing genes that are important in neutrophil development, which can really cause a lot of problems in terms of recurrent infections and infections that are worse and can lead to death.
And some diseases like cancer will cause neutropenia. And it's a real problem because when you don't have enough neutrophils, you become highly susceptible to lots of different bacterial infections.
Yes, yes. And so, I did a little calculation, that 1 billion number per kilogram per day. So I weigh about 145 pounds, and that's like 66.7 kilograms. So, essentially, I'm making 66.7 billion neutrophils a day. And the dogma for neutrophils, at least what I had remembered from when I was in classes, is that they really live for about 24 hours. They're a fast turnover cell. You produce a lot of them. The potential of them, the hematopoietic potential from the stem cells is about 60% of those stem cells are used, or at least are developing into neutrophils. But recently, there's been a lot of evidence that these neutrophils in tissues can last longer, particularly in the context of infection or disease, when they're getting a lot of signals from the tissues and or bacteria or of infectious pathogens. So it's kind of being challenged a bit that tissue neutrophils are going to be longer lived than blood neutrophils. And so,
yeah, I always call these guys the kamikaze pilots.
Yeah,
when I teach immunology, because they're really terminally differentiated. They don't proliferate that we know about, and they just go in and they do their thing and they kill lots of bacteria. But because they have so many of the effector mechanisms to deploy against microbes, they themselves are susceptible to collateral damage, and so they kill themselves or they die. And so everything we have, I don't know if you're going to go into this, but the pus that everybody has associated with infection, that's basically just neutrophil goop.
Yeah. No, it's a good thing to say in the beginning, because it is kind of this very visual, you can see pus, it's gross, you associate it with disease. And, and they also produce enzymes that sometimes can turn the color of the green. Yeah, make it that nasty green. And so, it's a lot of neutrophils. And if you can't clear that, if you're still getting these signals we're going to talk about for the neutrophils to keep coming, but your body's not clearing them, it can really be problematic. So a lot of neutrophils require a lot of regulation. And so in terms of their differentiation in the bone marrow from a hematopoietic stem cell to a fully mature neutrophil is really important in the regulation of that. So you would find them developing in the bone marrow after getting signals from cells that they are situated around. And so these potential neutrophils, they develop in these niches or niches. I don't know. Do you guys say niche?
Niche.
Okay.
Niche.
Kathy Spindler said it's niche. Yeah.
That's fine. I will go with Kathy Spindler. That's fine. And so these niches sit in between kind of where the bone marrow and the bone kind of transition. And so there are osteoclasts that lie in close proximity to these specialized niches, and they give these neutrophils or pre-neutrophil signals to develop, and we'll talk in a little bit about what that means in terms of developing granules that hold these powerful, as Cindy said, kind of kamikaze-like proteins that kill each other and things around it. And so these cells surrounding in the niche are expressing chemokines. And we've talked about chemokines. And so they're these chemotactic molecules that help create gradients to tell cells where to go. We've talked about them in the context of cells releasing from your thymus and having kind of these temporal associations with day and night. I don't remember exactly what episode that was. But it
might have been two.
Yeah, it was early. Gosh,
we've been early.
Just doing this for about a little over a year. And so, so chemokine, and the one that these numbers, CXCL12, and the CXC is just a motif within that chemokine to help us distinguish different families of chemokines. And so the ligand then for the receptor on the neutrophil, which is CXCR receptor 4, helps keep the neutrophil in this bone marrow osteoclast niche until it is then mature enough to leave the bone marrow and get signals to migrate into the vasculature. And so over time, once these neutrophils, they start to become more mature, they're going to lose the receptivity to CXCL12. And so then they are less tied down to that bone marrow, and then they can develop affinity for other types of integrins that might be expressed on other parts of the vasculature to help them move. So this balance between migrating out of the bone marrow is also regulated by something called granulopoiesis. And that is basically when the neutrophil is developing granules inside of the cell. And really neutrophils, I mean, when you're looking up the different types of proteins inside these cells, it's like over 700.
That's crazy. And they package them into different granules, right?
They do, different granules that develop in different times. So it's temporal based on what stage they're in. And so there's an appearance of primary, secondary, and band, basically then working its way from a myeloblast, which is you're not going to see that segmented nucleus quite yet. And those are the primary granules in the myeloblasts and promyeloblasts. So you have the large round nuclei at this point. But then the stage that follows that, where you get more specific granules and you start to see a kidney-shaped structure if you're looking at these cells, staining them, as we mentioned before. And then as it moves on to transition into what we see as this band nucleus, then they're going to have gelatinous granules is kind of the next transition. And interestingly, I think within the last couple of years, they found another transition called ficolin, which is it's like a lipid. It's a lipid that it's an innate immune lipid that can help decrease certain proteins. But they only found this specific development of the granule because it was a late stage granule. It didn't develop really until the neutrophil had become nearly mature. And how they found it was just by adding another Percol gradient. So how we spin out these granules is by different density gradients. And by adding a fourth density gradient, they were able to find this, I guess, new kind of phase of granule development, which is interesting. I always like to hear when things this is something we found within the last 2 years. There's just so much that we really don't know about a cell that's been studied since the 1800s when we think about the history of neutrophils. Yeah.
So do you want to talk a little bit about why we care so much about all these granules?
Yeah. What
definitely. Is it about neutrophils and neutrophil granules that are so important?
Okay, yeah. So within those granules, they pack a lot of different potentially harmful substances. And so when we we maybe could talk about respiratory bursts now. Is that what you're alluding to? And
you could, or you could just, I was just thinking that we could let the listeners know really why we care so much about the fact that these have all these different granules and they have different things packaged in them.
Yeah, sure. So, within those granules are when the neutrophil is activated and it secretes the contents. It does a lot of different things to the environment. So it can kill cells. It can degrade extracellular matrix to allow the neutrophil to move more easily. And it can actually also send signals to other cells to promote the migration of neutrophils. And we can go through all the different things that are inside, but in general, if we're looking at just why they have these granules, it's so they can really pack a big punch in terms of killing cells that are potentially infected.
And the other thing that's interesting about what you said is you mentioned a lot about their nuclear structure and the lobe nucleus and everything. And it's good to mention that we care about that because these are the easiest cells to identify, immune cells to identify in a tissue, right? And it's used characteristically and diagnostically because if you see these multi-lobe nuclei, then you know for sure you have this acute infection where these early responders have come into the tissue. So they're used diagnostically and they're really easy to identify in the tissue. And that's why we care so much about the structure of that nucleus and how it's so different from the other cell types.
Right. Absolutely. So yeah, it's a way, like you mentioned, clinically to diagnose and to see what's going on. One of the first killing cells that comes on the scene in terms of your immune response. So that's just introducing the cells. And so I'm going to talk a little bit about the migration of these cells, unless there's anything else we want to bring up. Just a general
That's
sense.
cool too. And that's really relevant to the papers are going to talk about, which are so cool.
They're so cool. Yep. Yeah, I'm going to bring up 2 snippets of a paper. The papers aren't long, and I'm not going to really dive into them because I just want to give a general overview. But the migration of these cells and coming out of the bone marrow and then into the vasculature, well, they have to have a way to exit the vasculature and go into tissues when they get the signals to do so. And so what really the signals are is damage done to tissues by a wide range of things. So tissue injury, pathogens of different kinds, both bacterial, viral, and fungal. And it creates this signal of a gradient of chemoattractants. And so when you have tissue that is damaged, it's going to release things like chemokines and cytokines that are able to bind receptors on the neutrophil and also bind endothelial cells. And the migration of these cells is highly dependent on endothelial cells on the vasculature, so the artery. When you have a neutrophil basally, just hanging out homeostatically, it may be polarized. What I mean by that is the nucleus may be polarized to one way or the other. Basal polarization can happen, but it's likely not going to start binding to the edges of the endothelial cells in the vasculature until it gets signals to do so. And so the receptors, there's a lot of receptors on the surface of these neutrophils that can sense these pro-inflammatory mediators to modulate migration and function. And really what that comes down to intercellularly are things called G protein coupled receptors. We talked a lot about some of these FC receptors in past episodes. And so I'm going to kind of elucidate in the next couple minutes how that can take a neutrophil in the vasculature. And then if you've heard the term diapedesis or extravagination, it's going to go through the endothelial cells to the tissue. And so the first signals that it gets are things called damage or danger associated molecular patterns. Now, we've talked previously about pattern associated molecular patterns, pattern associated patterns in terms of on the pathogen. Sorry. Yeah. Molecular patterns, thank you, on the surface of bacteria and viruses because they can be detected by toll-like receptors on the surface of cells. But your tissues secrete, when they're damaged, I mean, they are proteins that they're around in your body naturally, but when your tissue is damaged, they're going to secrete more of those. And cells in your body can detect these, the DAMPs is what I'll refer to them as, and that's kind of like the first signal to allow neutrophils to they're going to start to roll on the surface of endothelial cells. And, Cindy, when I think about these danger signals and how they're different from pathogen associated molecular patterns, it really can be, which one came first? So, did the pathogen cause the tissue damage that allowed these proteins, polysaccharides? There's a lot of examples, DNA, RNA, metabolites. These are things that are in our bodies anyways, but how our body's able to distinguish, it can be tricky in terms of what came first. There's a theory that I'm interested in.
I think you can have either one. Will trigger it. So there are instances where you have non-infectious inflammatory damage where you can have neutrophils infiltrating, like drug toxicities and things like that. And in those cases, you're not going to have PAMPs and maybe only DAMPs. We often refer to them in viral infections as well, because some viruses evade some of the immune responses, but yet might cause damage when they lyse a cell, for example, and release DAMPs. That's another example of where a DAMP might be the only thing pretty much that the immune cells see. But clearly, PAMP triggering could also induce the changes in the endothelium to recruit the neutrophils as well. And you were going to say you were going to mention the danger theory?
Yeah, it's really interesting. And I know it's come up in my classes before, but I don't know if you wanted to touch on that. It always has been fascinating to me.
I think it's really cool. Polly Matzinger came up with this a number of years ago. I remember she came and gave a talk when I was in graduate school, and I was just absolutely fascinated by this idea that the immune system could really be evolved to detect danger, because it doesn't necessarily need to know if it's a pathogen or not, right? Because and now we know years and years later that our guts are full of bacteria that we don't necessarily want to attack because they're good for us, right? So, there's some microorganisms that might be present, but we don't need to generate an immune response to. But then there also might be instances, like I mentioned with drugs toxicity, for example, where there might be the need for an immune response, but not necessarily a pathogen. So distinguishing between when there is or isn't a pathogen may not be as critical as determining when there is or is not damage that needs to be repaired. And I think there was also a couple of review articles that have been written similar to this, where they talk about really the goal of the immune system is to maintain homeostasis. However, it's disrupted from that normal situation. And so whether it be tissue damage, you fall and break your leg, or you bruise yourself, or whether there's an infectious agent, the goal is to repair the tissue and return it back to normal.
So are you going to give us some examples of DAMPs?
DAMPs. So my favorite one is DNA.
Yeah.
So I study TLR9, which recognizes DNA.
So so that DAMP can be a PAMP also, depending on what's happening.
It can, and that's the really interesting part, right? Because it can be associated with autoimmune disease when you recognize your own DNA, or it can be really beneficial to host defense.
Do you know of a DAMP that is not a PAMP? Oh, we're getting really weedy here, but
not a PAMP. You're putting me on the spot. I know there's some.
I was going to say, I'm sure there's some. I mean, DAMPs can be secreted metabolites that purine metabolites, which,
got it.
I mean, purine metabolites aren't going to be on the surface of bacteria, so they're secreted forms. And so that's where I think it gets tricky in terms of defining responses to DAMPs and PAMPs, and neutrophils especially, and then what response they have in terms, it can be a little complicated, but it plays into like what Cindy was mentioning, whether you have this sterile based inflammatory response or a pathogen associated inflammatory response. I mean, in the end, the neutrophil sees the same, it's reacting to similar things. It just is a matter of is the pathogen cleared? But if it's a sterile inflammation, it could be a sign of autoimmunity. So yeah, that.
And you talked long enough for me to think of one, which is HMGB1, which is a nuclear protein.
Oh, okay.
Yay, I'm rescued.
Yay.
Okay. And there are many others, I presume.
There are.
Okay, got it.
Yep.
So the things that shouldn't be coming out of cells, they should be in the cell. But if there's damage,
the important point.
Yeah, exactly.
Right. Or if they ever do see them, maybe they're, you bind a receptor once, but in a lot of their immune response, you're going to need a lot more. You're going to need a concentrated amount of receptors, and then they kind of co-localize on the surface to then bind to other things. And so maybe then it's a great, like we're mentioning, these gradients of molecules, and that plays a role as well.
Mm-hmm. So,
yeah.
And these things don't necessarily come out because of infections. It could be tissue damage, as you mentioned, right?
Yep. I always do an example where I'm teaching this and the students are starting to fall asleep, and I trip and I pretend to bang into something, and I was like, oh, see, I could have just gotten a bruise, and that's an example of a non-infectious tissue damage. But it wakes them up, they all laugh, and they remember that.
So I gave blood yesterday, and I'm looking at my arm, and it's all bruised, right?
Oh, no.
She messed up, so I'm thinking there must be some neutrophils here, right?
Probably, yeah.
Yeah, probably.
Although since it was yesterday, they may be gone already, right?
Yeah, if they 24 hours, but you're always producing them. 10 billion per kilogram per day. Yeah. So talking about, some of these things on the surface of the neutrophils, and so when they're floating around in your blood vessel, you have something, I mentioned diapedesis, and if you've heard of that before, many different cell types do this. Cells need to move from circulation to the tissues. But in the case of neutrophils, it's just really well studied, and a lot better defined, I think, than some of the other cell types. But your neutrophil, when it's rolling along the endothelial cells, there are things called selectins that are on the surface of the endothelium, and those selectins are upregulated when they get signals from activated macrophages and other neutrophils, but macrophages that secrete cytokines like TNF-alpha. And so this is all playing into activating endothelial cells so that they can better grab a hold of neutrophils as neutrophils are floating along in our vasculature, to then basically tell them the signal that there's tissue damage, there's something that needs to be killed, so you need to quickly move and get to that spot. And it's
Have you seen the in vivo imaging of the blood vessels with the neutrophils flowing through the blood vessels, and then when they add a stimulus, they attach?
Yes,
because
I don't have it here.
I wouldn't characterize that as floating by. I sort of think of that as like, sitting on an inner tube, floating around a pool or a thing.
Right.
They are flying by like light
speed.
Yes,
and you're right.
They need to attach to that endothelium, but they need to grab on tight.
Right. Yes. And actually, that's a good point, because when we talk about neutrophils migrating, you have to think about blood flow, because I think some experiments that maybe were done at what we can say is low shear stress. So shear is just the force that's exerted, upon either the tissue or the cells as blood is moving. And so low shear versus high shear. And yes, like you said, they're moving quickly. And so they have to have a concentrated amount of those integrants on the surface of the endothelium to be able to grab a hold of them, to start the rolling process, so that they can grab a hold of more receptors or ligands on the surface of the endothelium, so that they can become closely knit to the endothelium and go through. So yeah, and that's going to kind of lead into the little snippet that I've included, but it's capture rolling, where it's a binding to these p-selectins from receptors on the neutrophil, but that's a lower affinity, but it's starting to slow it down. And so after it slows it down, it has the opportunity, if there is danger in the area, possibly things like IL-8s are going to bind to receptors on the endothelium, and then consequently bind to receptors on the neutrophil as like a second signal. And then there's a third signal, which are these things called ICAMs and VCAMs, these basically adhesion molecules that bind more tightly to the neutrophil. So it's this process of adhesion, just making sure the neutrophil is adhering to the endothelial cells so that they can go through paracellularly. And so it becomes an adhesion and then goes to a crawling. And it's amazing, the neutrophils, and there's videos, but they actually flatten. I mean, they're so morphologically, they can change so much more drastically than T cells and B cells. And they're completely flattening themselves to then migrate through the vessels and then go into the tissue towards these chemokine gradients that can, like I mentioned, CXCL8 or IL-8, they can be soluble in the tissue, but they also bind to these aminoglycosides on the tissue and creating kind of like this, come here to me. This is, you see a little bit of me and you see more and more of me, and the neutrophil will bind to that. And that's really this gradient that I'm mentioning. So diapedesis, it's the theme of it, the concept of it, it's most well studied in neutrophils, but this is something that all cells have to do to migrate throughout tissues.
And these are disease associated. This is really important, this whole process, because if any of those interactions are disrupted, you end up with diseases like leukocyte adhesion deficiency.
Right. If you don't have the ability to maybe bind those first selectins that are on the surface, right, or maybe the secondary signals like IL-8 or PCAM, ICAM, you don't have the ability of your cells. And I'm pretty sure then that would clinically result in neutrophilia in the blood, because they can't migrate out of the blood.
That's right. And then they can't get into the tissues. And so if the body doesn't have access to those neutrophils, it sort of forms like a physical granuloma, or just the cells don't get into the tissues and the bacteria overgrow. So one of those two things is going to happen.
Right. And
but yeah.
Yeah. So the migration of the cells towards some type of signal, I mean, it's very relevant. You know, when I think of like Staph aureus, it's well known that they can they can induce these swarms of neutrophils. So that it's considered like, I guess they call it neutrophil swarming. And
I love the videos of that.
Yeah,
so cool.
It's cool. And maybe I only included the paper that I'm going to bring up, that video, but I'll try to see if I can find some more of these, because this visually, there's a lot of, I would say, engineering combined with biology, because there's a lot of these kind of microtubules that people put a force on to basically look like the blood shear force, and then look at these cells migrating through like some type of what do I want to say, like a glass tube essentially. And then you can take really nice video and really nice pictures of these neutrophils migrating. So yeah, I'll try to find some of those to include. But yeah, so these types of changes in the neutrophil, I mean, the surface receptors on the neutrophils, I had mentioned those G protein coupled receptors, and when they get activated, there's a whole downstream process of Syk kinases and activating actin reorganization, because when these cells flatten on the endothelial cells, I mean, they have to be able to very quickly mobilize their actin to be able to polarize and kind of form, and maybe that's why they have this multilobular nucleus. I don't know if that's been kind of defined as that's why they have this nucleus, but maybe it helps in terms of creating this very plastic cell where it can move and change shapes. So moving on in terms of the snippet paper, because it has to do with migration of the cells and how they move in tissue or from vasculature to tissue. And there are these things called tethers and slings. And tethers and slings, I would say, I didn't really find out about that until I was my master's when this paper came out, and I was really interested in neutrophils and the way that these microvilli that are on the surface of these neutrophils. So as they're rolling along, like what Cindy said, I mean, the force of blood is they're really quickly moving. But let's say that there's injury and there's a buildup of P-selectin. So P-selectin is in granules in these endothelial cells that's then brought to the surface of the membrane when it's seeing things like TNF-alpha and IL-8, that kind of these cytokines that are associated with inflammation. So if you have enough of those, and spatiotemporally, it's important to have kind of a congregation. They will grab a hold of the neutrophil, but they hold on to the neutrophil long enough so that there's then a force. So there's two different forces acting on the neutrophil, one pushing it back, which is these tethers, and then one pushing it forward, which is the blood flow. And that force, that eventually those tethers that are pulling back on these microvilli will release, and that will sling forward around the neutrophil, this sling that it's almost like a gecko, if you can picture a frog, right? I mean, it goes all the way, almost like I can't remember how the distance it can go, but there's a video I included where it can sling far in front of it to help pull it along the endothelium. And so this little snippet that I've included here, the first author, the title is Slings Enable Neutrophil Rolling at High Shear. And so that's the difference. Really what we're talking about is how can these neutrophils stop themselves and get to the edge, or flatten themselves enough so that they can stop and migrate through. So the first author's last name is Sund, S-U-N-D-D. The last author's last name is Ley, L-E-Y. I don't know. Should I go through all of them?
No, you don't have to. Just tell us where they're from.
Yes. So they're from La Jolla in California. Let me see. Yeah, La Jolla Institute for Allergy and Immunology in California. And so this paper, was published in 2012. And so they used these really neat flow chambers, very small flow chambers, and they would take neutrophils that are either naive or activated, and they would add them to these flow chambers. And they had this microscopy where they could look along the XY plane to try to take video and pictures of the neutrophils migrating, because you can coat these microfluidic devices with whatever selectin. They use P-selectin. I mean, you could use a multiple. I'm sure for a cleaner system, they just chose one, but really, biologically, there's a lot of different receptors on those endothelial cells that's going to help the migration. And so they took these pictures of these slings, and they are fascinating. And you can actually visually see, and they do a little kind of a drawn picture as well. It's like, gosh, even maybe a lasso that the neutrophils are slinging and wrapping it around their entire, the entire radius of the cell, and then slinging it forward to pull them along. So, using some pretty neat technology. And then they follow up with that. Another group, they hadn't shown, I think it was 2 years ago, they show this in T cells and B cells too. And that, to me, was pretty fascinating, because I don't think of T and B cells as being, I would say, able to stretch as far as neutrophils.
Yeah.
Right. But there's a bit of a different mechanism. I think P-selectin, the receptor on the neutrophils, has to be activated by some costylations that B and T cells don't have. So it is a bit different. But I don't know. Did you guys get to check out that video?
Pretty cool.
Yeah, I love
Very
it.
It's
cool.
amazing. So
Very
what's the
cool.
sling attaching to? P-selectin?
On the other side, yes. And it's really neat because they were able to see that the receptor for P-selectin on this like pseudopodia, or, the microvilli that's slinging, they're basically congregated. And so they've actually kind of migrated together so that like a gecko almost, so that when they hit the surface, they bind to the P-selectin receptor.
And then the neutrophil kind of rolls and this thing wraps around it, right?
Yeah. Completely wraps around it, which is really, really neat. Mm-hmm.
I sort of think of it as like, I think like mountain climbing or trying to scale a castle wall. You throw the hook up and then you pull yourself up.
Yeah, I guess,
right.
it's a way of rolling, right? I guess there could be other ways, but it's one way, right?
Yeah.
Yeah, definitely. Yeah. And exactly. I'm sure that it's one way that the cells are able to roll. And so it's just a little short snippet, but it's a neat if you're interested in kind of the mechanisms of how this works, and one way that these cells then, and I always, looking at that video, I wonder how that doesn't get caught up. I mean, I guess, why that it's wrapping around it. So why that wouldn't get caught up in the cell, in the extracellular matrix, seems kind of, I don't know, suspicious, but the video shows it works. So
what's the idea, that this happens over and over again, or just once and then the neutrophil goes through the endothelium?
I mean, the assumption is it happens more than once.
Mm-hmm.
That video shows it more than once. So, but there has to be a time when it gets, flat enough, and the signals are strong enough that the cells paracellularly move through the endothelium, which are plumper, and they're, vasodilation is also happening. You're getting a lot of factors that help increase the diameter so they can move through. So eventually, they're going to migrate through.
So the rolling is in part to keep the neutrophil on the wall, but also to let it flatten so that it can get in, right?
Right. Mm-hmm.
I mean, I just wonder how it detects the gap between endothelial cells, right?
Do you know that they can dive through cells too?
Through cells?
They can.
How do they do that?
I don't know, but I think that's the coolest thing.
And do they go in vesicles or just by themselves?
They actively go just through the middle of the cell and come out the other side.
It's remarkable.
Yeah,
I know.
I mean, it has. I would assume it receptor mediated, but then
I don't know.
Right.
It's been seen by EM, so transmission EM going through because mostly they're primarily going to find a junction, and then they use PCAM to kind of get to zipper their way through in between the two cells and maintain the endothelial junction on either side so that the blood doesn't just leak out. But they have been shown to dive through the middle of cells too.
Steph, this is kind of out of nowhere, but if you go back on the phylogenetic tree, we talked about going back to organisms that have different kind of antibodies, right? How far back are there neutrophils that would do this? Do you know offhand?
this type of slinging and tethering. Is that what you're referencing, or just neutrophils in general?
Just neutrophils that would roll on endothelium and pass through. How far back do you have to go?
Gosh, I mean, I know that the term phagocyte and phagocytosis, Metchnikoff, Elia, I don't know how to...
Ellie
Metchnikoff.
Ellie, thank you. I mean, that was discovered in marine sponges. So that's quite evolutionarily a long time ago. I mean, the rolling, I don't think we...
you have to have blood vessels, right?
Yeah, sure.
Right. And I mean, there's phloem. You do have fluid that they have to migrate through to get to the tissues. There's still some type of, lumen that they migrate. So, yeah, I think it's evolutionarily, conserved in terms of thinking about sponges versus now. Yeah.
And insects have some sort of similar type of phagocyte too.
Right. Yes. That I'm not so up on. So maybe if an insect person wants to email, they could come on and talk about that. But yes.
But I don't think plants would have something like this because I don't think they have patrolling cells, do they? I could be schooled on this, but I have no idea.
Yeah, I don't know. Vincent, you've talked to a lot of plant people in the past. Do you have any?
I don't recall, though. Like you said, hemocytes and insects, those would be patrolling, but or could be. But I'm not aware in plants. But I could be wrong. Let us know.
Yep.
Yep.
Okay, so I guess since we've worked our way through migration, I mean, I'm just going through kind of broad concepts of neutrophils just to give a little background. So, we can dive in more, but really, I mean, neutrophil, we could take a couple episodes on some of these processes. I'm breezing over some of the very, mechanistic intracellular proteins. Is there anything else you guys?
But they're best known for eating things, right?
Yes. So once they get to the spot where they're not only eating bacteria, but they're eating dead cells. I'm pretty sure they can eat other neutrophils. I know macrophages eat neutrophils. Can neutrophils eat neutrophils?
Mm-hmm.
Yeah, I don't know. I think so.
If they're dying, the signals on the surface would be similar. So, they are best known for being very powerful effector cells that can destroy through phagocytosis, degranulation, reactive oxygen species, which I mentioned before, and then these extracellular traps, which I will kind of have a little snippet. I think they're so cool. But in terms of like phagocytosis, I mean, really, that's just the ability to uptake particulates with a certain size that have, some type of that you can develop then like an endosome or a phagosome inside the cell. But how neutrophils recognize what's supposed to be phagocytosed, it can be a couple different ways. So either receptors on the surface that have opsonized. So we've in the past mentioned like complement receptors, lectin-like receptors. So basically those are the things like an antibody is going to bind to a FC receptor to flag certain pathogens, and that then can be recognized by neutrophils to then endocytose and eat that pathogen. So opsonic receptors, but there's also other non-opsonic receptors. I mentioned like lectin-like recognition molecules, and even though toll-like receptors, I mean, they're not entry receptors, they're sensors, but they also can help promote some of the uptake of either of bacteria and viruses. So the injured cells, they're going to try to phagocytose, they're sending out these signals, and these signals can really, in terms of necrotic cells, you're just spilling out your contents. And so that's when we talk about DAMPs, or those danger associated molecular patterns. Those are the signals that bind to neutrophils. So then they uptake those, whatever we're talking about. So whether it be a microbe or pieces of dead cells, and the ability of those neutrophils to do that, then also, like we mentioned, can be clinically relevant, because if you're if your neutrophils cannot clear, if they can't eat or phagocytose, then you have a buildup, and it's very hard to clear infections. And
they don't want to just eat because they're hungry, right? They want to eat because they want to kill whatever they're eating.
Right. I mean, yes. So, right. Yeah. So that's the goal, is to clear. I mean, because they are short-lived cells, so they're just supposed to go in, eat, but phagocytose, clear. And so then you can potentially regain a homeostasis by removing some of these danger-associated signals. And really that crosstalk is going to stimulate more recruitment of neutrophils, or if you have a decreased amount of neutrophils being recruited, then you're not going to have as many of these factors spilling out of cells. So once the microbe or piece of cell enters inside the neutrophil, there's different stages called the phagosome inside. And so between going from an early to a late phagosome, it joins up with a lysosome to form this phagolysosome. And that's really where some of this oxygen-dependent killing is happening. So respiratory burst, this is really creating superoxide anions that are going to be converted to hydroxyl radicals. And those hydroxyl radicals are very damaging to your tissue and to cells. And so if you have a huge amount of neutrophils that are undergoing this respiratory burst, you can really cause potentially a lot of damage. Of course, there are ways that our body has. There's other enzymes that can help decrease the effects of that oxidative stress and these chemical molecules that contain oxygen that are really highly reactive. And the conversion of that is really what then can cause damage. And under normal conditions, I mean, you have reactive oxygen species that are byproducts. But in terms of this, if you have an imbalance of reactive oxygen species that is increased, that's when it can be problematic. But your body has things like antioxidants, and there's whole industries that have sprung up over, ingesting antioxidants to decrease your reactive oxygen species. But whether or not that works, I think your body, it depends on what the situation is. But
that's why I always drink my red wine.
Yeah, red. Exactly. So, you have oxygen dependent killing, and in terms of, and maybe I didn't bring this up, but the mechanism is you have nicotinamide adenine dinucleotide phosphate, or NADPH, on the membrane of these cells, and that oxidase can catalyze the reduction of molecular oxygen to the superoxide anion. So that catalysis is really then what can help to kill and either would it or degrade, whether it be tissues or microbes. So,
and we know again how important that is, because if there's lots of mutations that can happen in that reactive oxygen producing complex, and when you have those, it causes a disease called chronic granulomatous disease, and so you can't clear the bugs again. So the neutrophils make it there, but then they can't kill the bugs. And so, so each one of these things is incredibly important. And when they're defective, it causes disease.
Right.
So I was wondering if you know what happens when they can't eat something, right? So there's things that are too big, right?
So they can actually they can, in terms of the neutrophil nets, is that what you're transitioning to?
Partly that's getting to the paper a little bit, but also they have frustrated phagocytosis. So typically their goal is to eat whatever it is, right? And then deploy these granules and things at whatever they've eaten. But sometimes things are too big to eat. And so then they'll discharge things outside so they can degranulate as well.
Yes. So in that degranulation, that when I was talking back about those granules, there's many different types of proteins that are inside, and they're packed with hydrolases and bactericidal proteins. And so they just kind of spill them. There's a regulated mechanism by how they do that. But yes, that's if it's too big, if they can express some of these primary and secondary granules to help degrade, if it's like bacteria, in terms of the membranes, or even extracellular matrix, if the bacteria is caught in that matrix. I think for Staph aureus, there's developed a way that in the extracellular matrix, they can kind of capture that bacteria and then kill it that way. So both eating it or phagocytosing it, or secreting their antibacterial products to kill it that way. And then there's another way that they can basically secrete what's inside of their nucleus, is they shoot what's called these extracellular traps. And these are
like
really
Spider-Man.
Yeah, these are really, really cool. They were discovered in the 90s as what was thought to be a different way of apoptosis or necrosis. They weren't really looking at it in terms of killing bacteria, but that was when it was first described as a biological process, but not until 2004 when Brinkman et al., they detailed the process of what they called netosis or neutrophil extracellular traps. And there's different ways they go about it, but essentially they wanted, in the second snippet I have, they show you with electron microscopy and immunofluorescence what this looks like. But the neutrophil, what it can do then is once it's activated, they can just let out all of the contents inside of the nucleus. And so that can be basically all of the granules as well. But in terms of the DNA, it's decorated with histones and antibacterial proteins. I mean, it just spills out of the cell. And the cell either dies or this is fascinating that they can stay alive after doing that. I mean, I guess they're producing enough I guess they're replicating enough DNA that they can shoot DNA out and still have enough. I didn't go into that process, where they stay alive, but they are trying to trap bacteria so that other neutrophils or macrophages could then clear it. And so that second snippet, I don't have the authors here, do I? No, let me pull that up. Oh, Brinkman, yes.
It's in Arturo Sierlinski's lab.
Yes. So that's at the Max Planck Institute. And if you take a look at those, you can see that activated, not non-activated neutrophils, they will secrete their contents, and they're able then to decrease virulence factors by Staph aureus or Shigella toxin. Now, the bacteria, of course, this is evolutionary arms race over time. I mean, they found ways then to degrade the neutrophil traps. And so, they're going back and forth. But I just remember when I learned about this, I thought it was so cool that they can basically vomit up their contents to try to immobilize bacteria. It's very cool.
Go ahead.
How do they come out? Are they in vesicles that are then bringing the DNA out, and they fuse at the surface and the DNA comes out? Is that known?
I don't think so.
No. I mean, from what I understood is that it's not like like they're nicely packaged from the nucleus and then they migrate. I think it's a very quick process, and it's very non-specific because there's so much stuff in there. I mean, even I was looking at some of the effects of the histones on the DNA that they're secreting out can actually be also antiviral in terms of influenza. Some of those histones are they help to neutralize the virus. And so, yeah, I can look into that more in terms of exactly how, but I don't think it's well described.
Okay.
I would imagine there has to be some, and I didn't think about this until just now, but when we package DNA, and I was just talking about this with my kid this morning, we were talking about how much DNA you can have in one cell if you stretch it out, right? It's meters long, right? And it's packaged into these little things, and all the nucleosomes hold it, and it's secondary, tertiary structure and everything. But it's I thought of it almost like a spring coil, right? If you release something, and I don't think we know what that is yet, it just kind of shoots out like a jack in the box. That's how I thought about it. But then I always thought that it resulted in death automatically. But I guess some of the newer studies are saying that those cells can survive that process. And I don't really know how that would work.
Isn't that wild?
Fascinating.
Yeah. So they define it as suicidal netosis or vital netosis. So suicidal means that cell dies, which makes total sense. But the vital netosis, I mean, it's independent of other signaling pathways like reactive oxygen species signaling pathways, but they just see a loss. So when they're looking at these cells, whether electron microscopy or just measuring DNA, they see a loss of DNA and plasma membrane. So they're losing a part of the nuclear or the plasma membrane. So I'm assuming because that's where the DNA is actually moving out. So you have to make room for that. But then after they release it, their lifespan is not affected. So it's interesting. Now, there's also a third type. So the nuclear DNA is shut out. But in this third type, you can actually see mitochondrial DNA is released instead. And so what triggers nuclear versus mitochondrial is really interesting. And also with the vital netosis, so then is that cell able to then phagocytose? Are there
no
more
idea.
functions it can perform? I think that would be really interesting to find out. So yeah, that snippet is just, if you were to check it out, you can actually see, there's no videos, but there's some nice pictures of the extracellular traps. And that's been repeated, and people are looking at exactly what is inside, how the DNA uncondenses, and what the exact mechanism of when a neutrophil decides to do that, or a phagocytose. And so, yeah, it's pretty fascinating.
I think that's a fascinating question, is how does it know which mechanism it's going to deploy, right? So I think it's easy to imagine if it's trying to eat something, and if it can't eat it, then it's going to end up just fusing its granules with whatever it's trying to eat that's outside. But when would it choose voluntarily to spit out these DNA nets, which presumably can be problematic, right? Because we started all talking about what's a DAMP, and a DAMP is DNA, right? DNA outside of the cell. We have apoptosis in a lot of cases. That's this quiet, non-inflammatory death, and it packages the DNA and degrades it really carefully so that it's not outside the cell. And we have DNases and things that are present outside of cells to prevent DNA from being able to access cells like macrophages, where they can trigger TLR9 and exacerbate autoimmune disease. And then B cells, it can induce production of anti-DNA antibodies, and you get immune complexes, and you get things like lupus. So having DNA outside of the cell like this in large amounts is not always a good thing. So there must be some signal that says, there's something bad enough now that you better deploy this mechanism, because the risk is worth the risk, right?
Yes. And I think that's one of the main open questions when I was reading, is that exactly like that decision between, I know neutrophils don't make decisions, but the signals that induce one or the other, and what context, and then in a lot of these, like you mentioned, autoimmune diseases like lupus, rheumatoid arthritis, and if they're inducing more netosis, that could be a big problem. So that is a very brief but overarching overview of neutrophils and some of the things they do. So, we know that they are produced in huge quantities in your body, both basally, but then exponentially when you're infected. They migrate out of the bone marrow, and they're migrating into blood. And then once they're in blood, if they get the correct signals by either chemokines or some of these selectins that are going to enhance expression when there's injury on the endothelial cells, they move through, they go into tissue to then undergo their effector function, which would be phagocytosing or netosis or degranulization of those primary, secondary granules that I had talked about earlier. And so I kind of avoided listing just protein upon protein, because that's not particularly interesting, but there is a lot of proteins that are involved. And so I had pulled up, there's a good review of all the different potentially disorders that can come, neutrophil disorders. And there's a lot. And some of them, being genetic disorders, there's no cures, and sometimes, it involves in recurrent bacterial infections and death. So it's very, very serious.
And there are some viral infections that are controlled by neutrophils. Flu is a well-known one, but there are others as well, but not as much as bacteria, I think, and fungi.
Right. I wonder too, because flu does induce quite a bit of tissue damage. I would imagine if the virus is inducing a lot of pathology, then that would recruit more neutrophils, and they play maybe a bigger role. Mm-hmm. But yeah, so, I'm not going to list all of the potential disorders, but I don't know, Cindy, is there something interesting about neutrophil disorders that you've heard about or had experiences with? I mean, cancer, I'm going to kind of bring up at the end, but could be quite devastating.
Now, I think we've touched on some of the major ones as we went through, chronic granulomatous disease, leukocyte adhesion deficiency, or genetic disorders that lead to, inability of neutrophils to complete their task. And so, it emphasizes the importance of these cells. Obviously we knew they're important for a very long time, because you wouldn't have as many as you do in your blood if they weren't doing something important, right?
Sure. Yeah. Yeah. And in terms of the subsets of these types of cells, when you think about pathology and cancer in particular, it seems that there are signals that certain diseases like cancer put out that will promote the premature migration of these cells out of the bone marrow. And so what you will see is increased levels of low density neutrophils, and they've actually, they're kind of their own subset of neutrophils, although I think there's some debate about whether they're an actual subset or of neutrophil with their own functions, or like I mentioned, they're just these immature neutrophils that have migrated too early, but they do seem to be increased during disease. And what those signals are, I think, is important, because then those neutrophils, if they're not fully developed, they can't perform their effector functions. And I think understanding how they come out of bone marrow early is going to be important in terms of cancer. I know there's a lot of work undergoing with that facet of this.
That was great. That was a lovely description of the many facets of neutrophils. I love those slings. I just think that's and the nets. You picked great things to talk about, because I think those are the two most exciting and interesting parts of what neutrophils can do.
Right. Oh, I agree. I got tripped up a little bit when discussing some of these reactive oxygen species, because I was looking at the notes thinking there's a lot of different chemical reactions I could go through and describe. Yeah. Sometimes when doing these big overview ones, that's maybe not the point. I think the point of this was just to describe a really neat cell type and some of the functions it does and why they're important, as kind of people were asking, I think, for, immunology overviews. And so hopefully learned a little bit more.
How would you deplete neutrophils from, say, a mouse? With some antibody to surface antigen? Yeah.
So there's Ly6,
yeah.
C and G, and then some are on certain cells only, and some are expressed on multiple different cells. So whenever you do an immunodepletion, you inject an antibody into an animal's blood, and what happens is the antibody will bind to the target cells, and then other cells will eat those cells by antibody-dependent cell cytotoxicity. And so it will deplete those cells from the flow. But with a lot of that kinds of approaches, things aren't always expressed exclusively on one cell type. So identifying a marker that's only on your neutrophil and not on other cells can sometimes be tricky. There's ways to do it, but you have to be careful interpreting studies like that, because the other thing is you can deplete easily in the blood, but not so much in the tissue.
Yeah.
Because antibody access. So sometimes you don't deplete everything you want to deplete, and sometimes you deplete things you don't want to deplete. So,
but I'm not aware of a genetic way to ablate neutrophils, right? For some other cell types you can do that, but not for neutrophils.
I think you'd be pretty immunocompromised.
Oh, I mean, in mice it would be interesting, but
yeah. You mean in people? Not
in people. In mice, experimentally, I don't think there's a genetic way to get rid of neutrophils, right?
Well, looking at this table so if you are deficient from elastase is how the neutrophils are able it's needed. So I'm at Jackson Labs. So homozygous elastase neutrophil express knockout. So ELANE. So it's that ELANE gene that's in people if they don't have neutrophils. So I think that, but like Cindy said, that would be very challenging mouse model if you're looking at disease. But I think that would be considered a neutrophil knockout if I'm looking at it.
So there,
maybe
just
that's a lab
made mice.
Yeah, there's I found one here.
There is a mouse,
Characterization of a novel neutrophil deficient mouse strain.
Yeah. Okay. But I guess it's easier to do when you have these mice in these really, really clean facilities.
Mm-hmm.
I think it would be harder if it was a traditional facility.
So they used Cree to knock out MCL1 protein, and that made neutrophil deficient mice. Cool.
Yeah. Cool.
All right, so it's possible.
Yeah, possible. So I think, that's all I have, but we could do a couple emails if you want. I think we have a few.
Sure. Let's do some.
We have a backlog of some, I think.
I guess we do.
Yes. Go ahead, Steph. Take the first one.
Yeah, let me get back. I was looking at Jackson Labs. Okay. So the first one, Sam writes, hello, TPH-5, Twix podcast host, 5th podcast in the family. That's cute. I wanted to come up with a clever name to refer to you by, but I came up with nothing. But since this is an immunology-related podcast, I figured I could just create a 3-letter abbreviation in which one of the letters itself was an abbreviation for something else, add a number, and I'd fit right in.
This is perfect. I love it.
Perfect. Yeah. Oh, awesome. Your podcast has recently discussed the phenomenon of joint first authors and occasionally joint second authors. Side note, I saw on Twitter somebody was complaining about joint six authors, which is,
oh my gosh,
I mean, ridiculous. I don't even know what to say about that. I recently came across this article, which has a single first author, but both joint second authors and joint third authors. I don't know the field well enough to tell if this paper is sufficiently interesting to warrant either a full episode or snippet, but the research highlighted in this paper, and he links, or she links, not quite sure Sam can, either, makes it seem like this is pretty groundbreaking. Anyway, I'd be interested in hearing your thoughts, however you choose to present it. As an aside, listening to this podcast and audio immunity, I think I said it right, has me inspired to take Prof. Mauschewitz immunology course here at Columbia. So I look forward to hearing Professor Racaniello's guest lecture on vaccines later in the month. Thank you for all the work you put into the podcast, Sam Matthews. Do you know Sam?
Well, I met him at the lecture, which was last month.
It happened.
Yeah, it happened, and I met him, and he's taking this is an immunology course.
He's a law school candidate.
Yeah, he's a lawyer. He's going to be a lawyer.
That's crazy.
Yeah,
Oh.
there you go. He's taking,
Cool.
and I asked him if he was going to take my virology course, but he said he didn't have room in his schedule.
I'm surprised he has room in his schedule for an immunology course.
Yeah.
That's amazing.
Good for him. That's awesome. Yeah, in terms of the joint first, second author, I mean, you guys can give me kind of a history, but I mean, this has been kind of increasing. I mean, it has been increasing. I think it's just the huge competitive nature of science and people trying, I guess, but if joint second and third authors is not going to matter on a CV. So I'm not quite sure what the point of that is.
I would agree. I think the first author, traditionally has done the work, and that's why we have joint first authors now, so that you can say, these 2 people or 3 people or whatever. And this is happening more and more because we have many big teams. Although, let's see, I have a guest blogger who wrote a blog post, which is coming out of Allergy blog today. And she wrote about a paper just published from Bonnie Bassler's lab, which has just her and another person. Huh. So there are still 2 author papers, because when I was a student and postdoc, that was the norm, 2 author papers, right?
Right.
And there's some people who publish with their pets too.
Oh, yeah.
Seriously?
Well,
yes.
Oh, wow.
Yes.
Polly, not Polly.
Yes.
But I
Dog
think
drama.
I would agree, and possibly we're going to get some pushback, but beyond first author and last author, which is, traditionally the PI, I'm not sure what joint second, third, fourth, fifth, sixth, etc. authorship would help at all,
Or mean necessarily.
Yeah. Now, I know that being the PI is important.
Yes.
I was criticized on a recent grant application for not being the last author on enough papers, in the last 10 years, which I think is just ridiculous because, well, I'm not going to go into that. But the thing is that you can be a joint first author, but there are different ways to spin that too, right? You could do it alphabetically, or one person could say, I want to be first author. I want to be first of the 2 joint authors, even though it's not alphabetical. Because I think I did more work than you.
Then it's not joint, right?
It's just, it happens.
Tricky.
Anyway, this paper is about basophils, which probably we'll do another time. And a basophil, that's our logo. The immune logo is a basophil. Another one of these fill cells.
Yep. And this paper that they reference is on IgD, which is the the black sheep of the immunoglobulins. We just sort of say, oh yeah, they make it at the beginning and then they switch out of making that, or, IgM and IgD. And we always ignore IgD. So clearly there's something to learn.
Yeah.
Cindy, can you take the next one?
I sure can. Jim writes, catching up on the Immune podcast and got through number 7 today. Loved this explanation of complement and its relationship to Alzheimer's disease. A little surprised at the pessimism about aging, forgetting words, falling apart physically, having the body wear out at young age, decreased libido. Don't think this decreased libido got mentioned.
No, I don't think so.
Alzheimer's seemed inevitable.
It's a family program.
He would like to offer a pick of the week about this stuff. The link is to a YouTube video by Joel Furman, MD, called Eat to Live. It may offer some hope for improving the chance of living in pretty good health with a well-functioning memory and libido. It has worked pretty well for us in the last 2 years. Improvement is gradual and there are setbacks. So I don't know what setbacks means. Is that relating to the libido or to the functioning memory? Or did both. They forget? I don't know how you measure whether it's working well in 2 years, if it's a long-term sort of treatment. I didn't watch the video.
So here's the problem, right? Unless you do a clinical trial, which we know is the gold standard for figuring out whether any regimen actually works.
That's correct.
Everything is anecdotal. So Jim, you may have found that it helped you, but that doesn't mean it's actually doing what you think it is. There are things we call the placebo effect, right? Which is 30% of any study at any time. So that's why we do clinical trials, and they're very carefully designed and managed. And that's how we decide whether a drug, a vaccine, a medicine, or any kind of regimen works. And it's really important to design it properly, and anything else is really anecdotal. But I always say, if you do something and you feel better, as long as it doesn't hurt, you do it, right?
That's right.
Now, I'm the one who jokes about aging because I'm the old one on this podcast. And I really joke about it. I'm not serious. I understand that all these things that happen are part of aging, and I accept it, and I'll never really seriously complain. The thing that I find really interesting is that lots of things happen as you age, and it's probably because we're living longer than our bodies can handle. Right. You know, evolution has designed an organism that really the main goal is reproduction, right? To make more organisms to keep the organism going. And so, most of us, if we get to 70s and 80s and 90s, things break because they're not designed to go that long, because you're not reproducing anymore. You have no contribution to the gene pool anymore. So that's just the way it is. And, just think, here's a hypothetical. What if everyone lived forever and were healthy? That's not beyond medical research at some point, but that would be a problem. The world would.
Our poor Earth could not sustain.
Yeah.
Nope. Well, you couldn't make more. You just have to deal with who's here, right?
Yeah, you couldn't reproduce anymore, but that'd be so weird. However, we're fooling around with things, as you know, and so
we are.
It's going to be very interesting. All right, well, let's do one more from Ken. Greetings all. Episode 9 included a discussion of introductory books about immunology. I recently read The Immune System, a very short introduction by Paul Klennerman, Oxford University Press. Since immunology is far from my field, computers and education, I like these short introductions because they don't take a long time to read. I also enjoyed Viruses, a very short introduction by Dorothy Crawford for the same reason. I haven't read the microbiology one, and it seems there isn't one on parasites.
Guess we have to get Dixon working on that, huh?
Well, Dixon, has a parasitic diseases book, which is not short. It's basically an encyclopedia, which each chapter is a different parasite.
Yep.
However, it is free online. And so sometimes free trumps short, right?
That's true.
And you can go to parasiteswithoutborders.com, and you can find a link to the PDF, which is being made available through donations, and they give away that, and they also give the same textbooks to medical schools globally. And so it's a very good textbook. All right. Let's move on to some picks of the week. Stephanie, what do you have?
Well, it's kind of funny. I was going to do something a little different.
You're going to sing?
Well, no, you don't want to hear me sing. So, I think one of the things that I hear a lot about trainees is there's a lot of things that come at you in the training process that you don't know how to deal with, and just opinions from senior scientists would be helpful. So I think I'm going to institute a little get your opinion on something and see what you think. So you don't have to tell your honest opinion if you don't want to. I don't know. This is new, but we're having we were having an issue with one of my papers where the journal that I submitted to, they will reveal the reviewers' names if it gets accepted. So if it's rejected, then they don't reveal the names. And so I guess they were having a hard time getting reviewers because of this or blah, blah, blah. So the question is, I was interested in your opinion, whether you agree with that philosophy of journals. Do you think that everyone should know everyone's reviewer? Or do you think it's better if it's anonymous? And that's kind of that opinion is just, I was interested to hear.
What do you think, Vincent?
I think it's fine if you know your reviewers. I know some people who have historically signed their reviews, so people know who it is. The problem with being anonymous is that people hide behind nasty comments.
That's right.
And I think that should go. I don't think there's any room for nastiness in anything, but particularly in science. Now, the reason I like this is because at least one journal, there are probably more, MBio, which is an ASM journal, has a publication track where you can get your own reviews of a paper. So you have a paper you want to submit to them. You could write a few people, say, would you review our paper? And you know who they are, obviously, because you're writing to them. They will send you the reviews. You modify the paper and you send them back to them and they say, I'm fine with this. And then you send the whole package to MBio and they decide whether to accept it or not. And it goes to an editor. And I've gotten a couple of these where I go, well, this is great because I don't have to send the paper out for reviews. I get a package of reviews and the reviewers are known. They say, this is fine. The authors addressed all the issues and it gets published really quickly. And I think that's great. I think there needs to be more transparency. I think the hiding behind, nasty comments, doing 20 more experiments. You would never tell your colleague, you need to do 20 more experiments. You would tell them the ones they need to do. But so I think we should move to that. I think publication has to change in a big way, and that would be fine. And I think the MBio experience tells me it's fine.
Cool. Okay.
What do you think, Cindy?
I tend to agree with that. There's some times where there might be a really tough review that you have to give, right? There's you really like a person and you like the work, but there's a real critical problem or something like that. And that's a tough one because you don't want to hurt people's feelings. So it's not necessarily hiding behind a nasty comment, but it still can be hard to hear if you have a critical flaw in design or something like that.
Well, you could say that in a constructive way.
Yeah, I think the constructive reviews are really helpful. I definitely try to do that. I don't give busy work, but I certainly have gotten reviews where they have asked me to do it 7 different ways to get the same answer. And I said, all this is doing is holding up the publication of the study.
Yeah.
Sure.
I mean, the comments I would like to avoid would be, this work does not advance the field significantly. Yeah. I mean, that's totally unnecessary.
Right,
okay?
right.
Just tell us if the experiments justify the conclusions or not. That's all.
That's right.
And so the problem, this gets to a bigger problem, which is, of course, there shouldn't be this hierarchy of journals. Science should be journal agnostic. You should look at a work and not where it was published. And I see people on Twitter all the time saying, oh, I just got a Nature paper. I just got a Cell paper. Really, what was the actual science? Isn't that more important than telling us that you got this or that? Of course, I don't say that, but
I know, but I'm coming from a hopeful, I would say that it just has to do with, my ability to get a job.
I understand.
And unfortunately, it's the senior. I mean, if senior,
no, you're going to keep doing this.
I know.
And it doesn't stop because it's your ability to get a grant too.
Totally. But at some point, this is going to change very slowly. Right. And my pick has something to do with that. But I see signs of change. It's not going to happen overnight. And you're going to keep wanting Nature Science and Cell and all that. But at some point in the future, it's not going to be a journal anymore. It's just going to be published somewhere. And I think that's the way it should be.
Great. Well, I'll go back to a more traditional pick of the week next time. But I just
no problem. That's
value
good.
your opinion.
It's a good idea.
Yeah.
Cindy, what do you have?
Mine's a little bit heavy because I have a very close friend who just relapsed with triple negative breast cancer. And the prognosis is not good. And I asked her, I said, I'm doing this podcast on immune. I said, I'd like to make a tribute to you. I said, what would you choose as a science website or a foundation or something that is of interest to you? And she chose the Breast Cancer Research Foundation. And she says that they really are raising money and giving it directly to research. And I looked it up, and they have it's a non-for-profit. And they have raised $569 million to support clinical and translational research. And they fund over 200 researchers in 6 continents and 13 countries. And they are at the forefront of supporting some of the more cutting edge technologies for breast cancer treatment. And so if you are feeling generous around this holiday season, I'm not advertising necessarily for them specifically, but she's really thinks that this particular foundation does a good job of getting the money to the researchers. So if you know people who work in this area, they can get research funding from the Breast Cancer Research Foundation, or they could donate if it's friends and family.
All right. I just gave $100.
Oh, that's so nice.
Thank you.
I'll post it on my, separately on my social media.
Thanks.
My pick has something to do with publishing. It's an article in The Scientist called Plan S, the ambitious initiative to end the reign of paywalls. So some of you may have heard that a few European funding agencies recently said, if you use our money to support your research, you cannot publish in any paywalled journal.
Wow.
And that's great. I mean, it's about $7 billion worth of research support.
That's awesome.
And of course, the big journals started screaming, you're going to ruin publishing. No.
Of course.
No. Right.
You're going to ruin your profits, but not publishing.
Exactly.
This is horrible that these journals just see profits and not publishing the science. Anyway, there's now additional people who have joined this, and there's a coalition. It's Coalition S. It's a group of 11 national agencies across Europe. And now the Wellcome Trust and the Gates Foundation and one foundation in Sweden have joined this coalition. All right. So it's growing, and eventually you'll get lots of the big funding, and then it's going to have to spread to the U.S. as well. And that's a problem because most of the big journals are not open access. They all have a little open access journal just to say, oh, we do open access.
Right.
But that's
right.
That's nonsense because their main journals are still paywalled.
Well, and it's very expensive to publish in those open access.
You do have to pay. Yes, absolutely.
Right. I mean, even more, I think more though than the non-open access.
Yeah, I think, PNAS, if you want it to be open access from day one, you have to pay additional, right? It's expensive. Mm-hmm. And, the funding agencies are going to have to pay for that. I think it's a good first move.
Yeah.
Eventually, it should not be upon us to pay for publishing our research. I think that's another ridiculous aspect of publishing, that we have to pay. I mean, can imagine you write a novel and you have to pay to get it published. It's crazy. We shouldn't.
Well, some people do that, but
I know
they publish their own.
Well, so you can self-publish, but it's usually you don't have to pay for it. And most of them are on demand. So if someone orders your book, it's printed and sent to them, or they can get it electronically. But I don't think we should have to pay to publish. There are ways to publish without having to pay. And when you pay nowadays, where I heard today, for example, that PNAS is no longer, starting in January, no longer going to publish a print version of the journal, which is great. And they're also no longer going to enforce page limits because they've had 6 pages is the limit of a PNAS paper. Now you could do whatever you want. It shouldn't cost a lot of money to publish anymore if it's all online. The reason you're paying a lot of money to Science and Cell and Nature is because they have all those other stories, the news stories and the commentaries and all that they pay for. That's not about science publication. Anyway, so that's my pick, is this interesting initiative to end the reign of paywalls. And that is Immune number 15. That's the last one for 2018. Next one will be 2019. You can find it at Apple Podcasts, microbe.tv/immune, many other places, by the way. We are now on Pandora. Well, we will be on Pandora. Twiv is on Pandora. I see that we could get podcasts on Alexa now, so I'm going to look into doing that. But most of you probably listen on your phone or tablet using an app. If you use an app, please subscribe. It really helps us to have the numbers of people who are listening, and that way we can do more things. So subscribe, and you'll get every episode automatically, and we will learn how many people are listening. If you really like what we do, please support us financially, microbe.tv/contribute. And of course, questions or comments, immune@microbe.tv. Cindy Leifer's at Cornell University. She's on Twitter, Cindy Leifer. Thanks, Cindy.
Thank you, and have a great holiday. See you in the new year.
Steph Langel's at Ohio State University on Twitter, Stephanie Langel. Thanks, Steph.
Thank you. This is great.
And that's the last time we will say you're at Ohio State University.
That's true. A transition.
Yeah, that's cool.
It's cool.
I'm Vincent Racaniello. You can find me at virology.ws, Twitter, PROFVRR. Happy holidays, everybody. Happy New Year. Music on Immune is by Steve Neal, SteveNealPercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.